Now we're going to go on to something else very interesting, but I think in quite a different style. Now we're going to talk to Sandy Chan, and I don't think she'll be singing. No. Okay. So no need to change for your dancing shoes. That will be later during the networking break. And there will also be alcohol, and us Nordics do better with dancing when we've had a little bit to drink, right? But Sandy is going to talk about water, because Sandy is something as fancily as a PhD in applied microbiology. I mean, I'm impressed already, and I haven't even heard her talk yet. And she's going to talk about that when we have this glass of water here, what is it really? What's really in there? So please give it up for Sandy Chan.
Let's see. Okay, so I got the invitation to this conference, the bridge, from my communicator, Nina, at Sweden Water Research, telling me, oh, this is a great conference, we need to go here. So thank you for the invitation, this is lovely. As a PhD student, and quite young still, I would like to say, and maybe a little bit naive, I really believe in the way Sweden Water Research work. We need to create platforms to create bridges between science and business. And I would like to show you, by showing you my project, how we did that, or doing that actually. My research side belongs to Lund University from an institution called Applied Microbiology. Here I have on the machines I need, the lab, and also senior researcher, professors with all the knowledge and experience that can support me. On the other side, I'm employed actually at the drinking water producer here in Skåne. It's called Sydvatten. They produce high quality drinking water for example, us here today. And here I have full access to all the treatment plants. I have also knowledge and experience from people working in the plant over 20 years. And I also belong to a research company called Sweden Water Research, which I represent today. Sweden Water Research was formed by 3 other companies, NSVA, VA Syd, and Sydvatten. NSVA and VA Syd are responsible of the drinking water distribution network for the west of Skåne, and they also take care of the wastewater for the cities. So these 3 companies wanted to collect all the research into one part for better collaboration, better communication, and also better research. So the goal of Sweden Water Research is to develop new, effective solutions to meet the future challenges facing the water service industry. So this morning's theme was clean water for everyone. And I really believe in this, and I really think that we should work together to reach this goal. As an engineer in the background, we really like to define stuff. We define this variable, we define this parameter. So I will ask you all, what is the definition of clean water? Please, just give me some words. Nothing? Drinkable? Safe? Safe? Yeah. I work daily with microbes of all kinds, and I have noticed when people are talking about that something is clean, usually they mean without particles, for example, no dust on my bench, or they mean no color, no stains or anything, or without germs. I agree that our drinking water should be without color, should be without particles, and should also smell good, or don't have any smell. But I think we should be a little bit careful when it comes to water without microbes, or sterile drinking water. And this leads to my topic of this masterclass, the invisible world in a glass of water. The world I'm talking about is the world of microbes. More specifically, about the bacteria that is in our drinking water system. I can tell you right now that the water here in Lund, for example, is far away from sterile. Far, far away. I will give you some numbers later. Actually, yeah. So I would like to open up this world for you, and hopefully you can see a tiny part of it. But before I can do that, I need to remind you all about this future of our water. This picture looks a little bit dark, I know, and a little bit sad maybe. And that's what we are facing when it comes to water. According to World Health Organization, due to climate change, we will be forced to face many challenges when it comes to drinking water. For example, due to increase of water temperature and decrease of water flow, we will have an increase of nutrients, we will have increase of water color, and increase of pollutants. And that will cause severe consequences when it comes to our drinking water. Another example is that we will have increase of sea level in some part of this world, and that will increase the risk for seawater contamination into our groundwater, making it unsuitable to produce drinking water. And as said before, increased nutrients in increased water color, which is basically increased organic matter, means that we will give the microbes in the water more food. Okay. So the reason why I want to talk about this topic today is, except for the future, is also that microbes are really small, they are basically invisible, and we humans tend to not care about things we don't see, unfortunately. But I will try to show you that they are small, they're invisible, but they play a very important role when it comes to drinking water. I will give you some clear example why we should care. And first thing, when I talk about, I work with microbes, people think, wow, these are those things that make us sick. And when it comes to water, of course, waterborne diseases is one of the reasons why we should care about microbes in the water. I will give you 3 examples from different outbreak stories and see what we can learn from these 3 stories. The first one was in Walkerton, 2000. Walkerton is a city in Ontario, Canada, and had during this time around or less than 5,000 people living there. During this outbreak, 2,500 people got infected by this pathogen, and 7 people died. So what happened? How did the pathogen, in this case E. coli, EHEC, the pathogenic one, enter the drinking water system? So during this time, now the contamination source came from a cattle farm. And it came due to a drinking water well that was located really close to this cattle farm. So the pathogen managed to enter the drinking water system by this well. It had been already reported since 1978 that this well, due to its location, was not suitable or should be monitored more closely for drinking water production. Unfortunately, this was not followed, and this was together with a combination that the operators at the water supplies didn't add enough chlorine into the water, caused this outbreak. So this is really an example of how lack of respect and lack of understanding of our drinking water system can cause this huge consequence in a society, and also 7 people losing their lives. My second story is from Östersund, which is here in Sweden, located here. We are here, so quite north up. So this pathogen in this story, Cryptosporidium hominis, is a parasite. It's an eukaryotic cell, so it's larger than most of the bacteria. So during this outbreak, it began with that the hospital started to receive a lot of phone calls from people living in Östersund with all symptoms from Cryptosporidium. It took 4 days from those phone calls before the government went out to recommend the citizens to boil their drinking water. And it took even 4 more days before they were able to confirm that the pathogen, Cryptosporidium, have contaminated the source water and also all the drinking water in Östersund. Quite bad. And the government didn't remove the recommendation of boiling the water until 3 months after the outbreak. And that was after the installation of the UV at the treatment plant and after a lot of flushing of the whole water system. In total, 27,000 people got infected, and it costed around 140 to 220 million Swedish krona, around 40 to 22 million euros. That's a lot. And the largest cost was due to production loss of production, due to people got sick, due to sick leave. And unfortunately also, 90% around 90% of this cost went to so-called short-term solution. For example, for the sick leave people and also for flushing the pipe system. And only a small part went for prevention of the future. My last story is quite interesting. It's almost like a detective's work. So in 1969, in a college in Massachusetts, 90 out of 97 football team members got infected by hepatitis. They couldn't find any common source with other students at the campus, so they started to think, it must be the training ground that is the contamination source. But how did this virus enter this teenager? By the training ground. That's really weird. So this training ground was located on the top of the college area with a pipe system having its end there with a drinking tap for the football team members. And during this way, we have some irrigation taps for the fields. And we also have a city down here. So what happened? It showed that some children with hepatitis have been playing close with the water from the irrigation taps, and almost during the same time, it had been a fire in the city. So this led to the conclusion that the children contaminated water around these irrigation taps, and due to the fire, a negative pressure had been built up into the pipe and drawn the contaminated water into the pipe system. So when the teenagers later, next day, had training and drank from the drinking water tap, the contaminated water had traveled to them in a very unfortunate way. So these 3 examples show us that how dangerous, how costly, and how complicated microbes can be in our drinking water. But they can actually cause other kinds of problems too when it comes to drinking water. For example, in drinking water distribution network, in pipes we have all kinds of microbes, and they can produce something that's called biofilm. It's a sticky material. So we actually have that on our teeth when we wake up before we brush our teeth. It can be a little bit sticky. That is biofilm. So they can produce that in pipe system too. And here, they can start to build a community and live and grow. And they can grow so much, so they can cause loss of flow rate, loss of pressure drop, and even clog the whole pipe. And some microbes in this biofilm can also, for example, Ferrobacillus, they can oxidize iron in the pipe material, causing it to leak. And there's also all kinds of microbes that can produce all kinds of acids. For example, sulfuric acid produced by the bacteria called Acidiothios bacteria that can make the pipe leak. And this costs a lot for society to dig up the pipe and replace it. That's really complicated and costly. So of course there's a reason for us to understand this world of microbes. And now I have been talking about the bad microbes in the water, the pathogens that can make us sick, and also the spoilage microbes that destroy our pipe system. But the majority of the inhabitants in the water are actually not pathogens, and they are not spoilage microbes. They are so-called the natural flora in our drinking water. I will give you some numbers. For example, in a glass of water, we have around 1,000 to, this is not working here, 1,000 to 100,000 bacteria per milliliter. In Lund, we have around 200,000 bacteria per milliliter, and that is quite a lot. That is around 40 million in a glass of water, which corresponds to almost like all people living in Spain, the country Spain, in a glass of water. That is the word we are talking about. And when it comes to a distribution network, that is even more. On the pipe surface, we have around 100,000 to 100 million bacteria per square centimeter. That is a lot. So what are they doing there? They're not harmful. They're not pathogens. They don't destroy the pipes. What do? We think that these are actually the good microbes. These are those that can actually outcompete the pathogens. They can also make the water better. So by having a good flora, we can keep the quality or even increase the quality of our drinking water. Unfortunately, very little is known about this natural flora because they're very hard to study. But I will try to show you a part of this from one of my projects in a treatment plant here. But before we can talk about that, I need to ask you another question. How do we measure microbes in water? We can't see them. How do we do? Any microbiologists here? No? I'm alone. And I will give you 3 examples. This one is plate count. It's over 100 years old, and you just add your water samples in there and let it grow for some days, and then you can count how many colonies, dots, we have on the plate. A newer method called flow cytometry counts all the bacteria in your water based on DNA. No growing, but only based on DNA. And then we have the third method, a very modern and complicated method actually, called next generation sequencing, where we sequence all the DNA in a sample to get the name, basically, of the whole flora. Which method do you think all the regulations are based on? Yes, this is a really leading question, but yes, plate count, the method that is over 100 years old. Let's talk a little bit more about them. So these plates are actually from my lab. These are also called MCF plates. Here we can grow, you select E. coli and coliforms from this. So the blue dot here is, this one is an E. coli, and the rest is coliforms. So this method, basically what you do is that you give food and nutrients to the group you're interested in, that you want to follow, and then you also incubate it in a temperature that they like, in this case 37 degrees, and then you wait, and then you count how many that have grown from your sample. When we are talking about the natural flora, the whole flora, and want to measure that, we reach the limit of this method, because we always have a selection. The closest thing you can come when it comes to the whole flora with plate count is to measure the heterotrophic bacteria. Standard method is to grow them at 22 degrees, which is, there we have a selection. For example, here in Sweden, we almost never have 22 degrees in our water. And we also select bacteria that likes to live on surface instead of swimming around in the water. So you lose quite a lot, and actually studies have shown that you can only measure around, less than 0.1% of all bacteria in your drinking water when you use plate count. So to not limit ourselves in our lab, we use DNA methods. We don't grow, yeah, we do to compare, but we don't use it so much. We use DNA, because all bacteria has DNA. So the two first methods, next generation sequencing and qPCR, here we measure one specific gene, the 16S ribosomal rRNA gene, which we use for taxonomic determination of bacteria. That is quite common. And here, since it's DNA, we can measure all of the bacteria. We don't select, we don't have a growth step, we don't have anything, just the DNA. The third method, flow cytometry with DNA staining, is quite different compared to these other two methods, because here we measure the DNA inside the cell directly, which means that we don't need to extract the DNA, which is quite costful and it's quite complicated to do. So I thought it's supposed to be a deep lecture. I heard that, I got that from the bridge. So let's go deeper into this method. We start with qPCR. So here, qPCR, we want to measure the 16S rRNA gene, which every bacteria has. So what we do is to take the extracted DNA already. We split them by basically just heat it. We heat it up, and then the DNA strand will just fall apart to two strands, two single-stranded DNA. And then we add something called primers. They are small, single-stranded DNA strands that only binds to the 16S rRNA gene. It can only bind here. So now we have a tiny part that is double-stranded. And here, our enzyme can start to bind and start to copy this strand. So it goes to this nucleotide. Let's say this one is at A, then it will add a T to this one. Next one is a G, then it will add a C, and so on. And then it will make a lot of copies of this specific double-stranded DNA that we are interested in. This enzyme is actually quite similar to an enzyme we have in our body that we use to copy our DNA to produce new cells. It's the same mechanism, but we just apply it in the lab. And then we add fluorochromes that binds to the double-stranded DNA and give us a light. It will fluoresce. So when we have a sample with a lot of bacteria, which means we will have a lot of this 16S gene, we will get a lot of double-stranded copies, and we will get a lot of light. And then when we have a sample with less DNA, less bacteria, less DNA, and then we will have less light. And actually, you can also tweak this method a little bit. You don't need to measure the 16S RNA gene to measure all the bacteria. Right now, in my lab, we are developing another 16S method, which targets another gene. The gene is called MLRA. You don't need to remember that. But this gene encodes to an enzyme that is capable to degrade algae toxin. So then we will have a method that can measure bacteria that can degrade algae toxin, for example. The second method we use is next generation sequencing. It's actually quite similar for qPCR. It's coming from qPCR. But instead of having this fluorochrome that binds to double-stranded DNA, we have the fluorochrome attached to the nucleotides. And each nucleotide gets its own color. So A gets red, for example, and T yellow, and so on. So when the enzyme comes here and starts to add the nucleotide, one by one, it will give light. First, if it's an A, it will give a red light. And then if it's a T, it will get a yellow light. And then we measure that. So then we know the order of the nucleotides that is coupled to the DNA. So the raw data will look like something like this. So this is the DNA, basically. And then you need some bioinformatics and computer skills to basically translate this to the bacteria name. And this is done for all of the bacteria in your sample simultaneously. So you get a lot of data, but then therefore you get the whole natural flora. The third method we use is flow cytometry with DNA staining. This method is, compared to the other methods, really fast. You don't need to extract DNA. And it's really easy to use and doesn't cost so much except for the machine, of course. But you don't need to extract the DNA. You don't need to have so advanced computer skills. And you can run it quite easily. So instead of extracting DNA, we stain the DNA with the fluorochrome. So what the flow cytometer does is that it actually analyzes one cell at a time in a flow, and then measure the light that comes out from the fluorochrome, from the bacteria, simultaneously, all the time. And it's really quick. From sampling to results, it takes only some few hours. At the lab, at the treatment plant here today, close by, they use heterotrophic plate count, the one I talked about before. And that one, it takes 3 days. The incubation time is 3 days. So they need to wait 3 days before they know if this water is okay or not. Compared to this method, that first of all measure all of the bacteria, not only the 0.1%. Secondly, it takes only some hours. So this is really a method you can apply into the industry, and that's what we have done due to the collaboration between Sydvatten and Lund University. So let's use this method to go through the local drinking water system here west of Skåne. Oh, I forgot this slide actually. You get even more except how many bacteria you have from your sample from flow cytometry. You also get something called fingerprint for each water. So this is quite interesting. So you see I have 4 different drinking water, 4 different water samples. The blue one is from a lake called Mälaren here in Sweden. The red one is a lake called Bolmen, also in Sweden. And the blue one is the tap water here in Lund, more specifically from Kemicentrum at Lund University, where I have my lab. And then we have the light blue one, which is from a bottled water from the brand Loka here in Sweden. So you can see that each water sample has its own fingerprint. And it's actually not so complicated. It's just the distribution of the DNA we have here for each bacteria in the flora. So when bacteria that is on this side has a lot of DNA in it, so-called high nucleic acid group of bacteria. And then on this side, we have bacteria that doesn't have so much DNA in it. It's called low nucleic acid bacteria. So let's use this method to go through our drinking water system. So Sydvatten has 2 source water that they use and also 2 drinking water systems. We have Lake Bolmen, which we take our source water from and transport it by a tunnel into the treatment plant called Ringsjöverket, which you can see here on this picture. And then I will stand here instead. And then we have another lake called Lake Vom, which is here, that we take source water from into the treatment plant called Vomverket. And this is the picture of Vomverket. Here is Vom Lake. And then these 2 treatment plants produce drinking water for west of Skåne and distributed from Helsingborg to Lund to Malmö and more actually. I just want to show you the tunnel. This is the tunnel from Bolmen to Ringsjöverket, and it's around 82 kilometers long, and this is not during when we use it, of course. This is during a renovation actually. But when we use it, it's actually totally full. So it's huge. So this is the process line of Vomverket. We start with the source water, and then we pump it into something called infiltration ponds, which is also called artificial groundwater recharger. So it's a biofilter. And then the water goes through these filters, goes through the ground around 60 meters, and then we pump it up again into the actual drinking water plant. We have some irrigation, we have some softening step, flocculation, a rapid filter to remove the flocculant particles, and then we have a water reservoir where we add chlorine before we pump it up to the consumers. So we went to this treatment plant and went through it with flow cytometry because it's so rapid, and then we see all the bacteria in our drinking water system at this treatment plant. So we start with, so many zeros, 7.2 million of bacteria into a treatment plant. That is what we have to deal with. And then after the infiltration step, we only have 240,000 left. That's quite impressive. And then, nothing really happened with this number. It's quite stable out. And as I said to you before, it's also the same number in a tap. It's around 200,000 bacteria per milliliter. Quite a lot. So when we look at the fingerprints, as I said before, when you have a change of a fingerprint, according to flow cytometry, you have a change of the natural flora. You have a change when it comes to the inhabitant in your water. Here we see that raw water, the red one, looks like this, and then after the infiltration ponds, we have a huge change. It shifts the whole community into something else. But then it's quite stable throughout the whole treatment plant. And that is actually a little bit expected. We have softening, we have irrigation. That doesn't really affect bacteria. So that was nice to see. If we go to another treatment plant, this is Ringsjöverket, which is the one that produces our drinking water, most of the drinking water for us here today. It looks different. We have flocculation first, and then we have some lamell sedimentation to get rid of the big particles, and then we have a rapid filter to remove the small, fine floccal particles. Then at the end we have a biological filter again, but these are called slow sand filters. And then we pump it out. Or we add chlorine, and then we pump it up. So this is the count. We start with less, actually, only 2.2 million cells per milliliter, compared to 7.2. Maybe the tunnel is actually doing something. We are not sure. And then we have a drastic reduction of the flocculation to only 270,000. And then it's decreasing a little bit, but not so much, to only 210,000 going out to the distribution network. If we look at the fingerprint, it looks quite different. It's changing a lot. We see it's changing from source water to lamell sedimentation, and not so much from rapid filters. And then suddenly, we get a bump here from the slow sand filters, from the biological filters, which is quite interesting. So let's dig a little bit deeper into this process step, this specific one. So I want to introduce slow sand filter by history. So slow sand filters was first used in 1804 to actually produce water for a bleachery. And then the leftovers water was then later sold to the people. And it was not until 1829 slow sand filters was used for production of public water in London. But during this time, we didn't know anything about pathogens and microbes and this kind of stuff. We only knew that, oh, but slow sand filters are good because they can remove particles from the water. That's it. It was not until 1892 we first saw the full potential of slow sand filters. During this time, we had two cities, Altona, in this red square almost, and Hamburg. These two cities took the drinking water from the river Elbe. And Hamburg didn't really have any treatment step, it only had sedimentation, compared to Altona that filter all the water from slow sand filters. And when this river got contaminated by cholera, 1/3 of the population in Hamburg got infected, and around 8,000 people died. And Altona, all the people in Altona managed to escape this tragedy, almost unharmed. Year after, Hamburg installed slow sand filters. So I show you that slow sand filters can be really useful, and it's still the most useful all-around water treatment step. It has some mechanical filtration, it has sedimentation, but also the most important one, biological cleaning, which is the biofilm I talked about before. Now we are using it. It's on sand particles, and this biofilm can remove all kinds of contaminants from our drinking water. It can remove organic matter, nitrate, pathogens, phosphor, and you name it. So we wanted to study these slow sand filters because almost nothing is known about them. So we look at the washing procedure, which is a common procedure we do at the treatment plant to clean this filter. We need to do that like 2 to 3 times per year. And what we do is actually, we just empty the filter. You see, this is a working filter. This is a filter that is getting washed. What we do is that we empty the water, scrape off 5 centimeters of the sand, and then we add the water again. And then we use it again. Quite easy. But this is a disturbance for the biofilm, for the community living in the sand. And we wanted to see how this affects the slow sand filters. So we took 3 slow sand filters that were quite different from each other. They are different because of some other parameters, actually. We have this one is a new one with only new sand. We bought it from somewhere else in Sweden and put it in a basin and started to use it. And then this one is another one that had mixed sand, new sand, like this filter, but then we added some old washed sand and mixed it and put it in. And we compared these two with one of the well-working slow sand filters that only had old sand that had been running over 20 years. So we look at them, and we started to look at them with the conventional microbial parameters, which is plate count. And this is coliforms we're looking at. Coliforms is an indicator organism. They are by themselves not pathogenic, so they're not harmful, but if you have them in your water, it's an indication that you have a contamination from either wastewater or ground or rainwater or birds. That is usually our problem, because the slow sand filters are open, so the birds like to swim there and play there, unfortunately. So you see, the red one is the ingoing water, which is open. So sometimes we have some increase of this group of coliforms, which is a warning system, not good. But then outgoing water for the well-established one is always 0. Somehow this filter managed to just remove these group of bacteria. And when we compare that to 2 other filters, it looks different. These 2 new is not behaving so good. You see, when we have an increase in the ingoing water, we also have an increase of the outgoing water. The green one is the outgoing water. And it's even worse for the new one here. You really see a peak here in the ingoing, and then you see a peak in the outgoing. Somehow this filter is not able to do this shift, like this one. So we follow that with flow cytometry and see, can we see that in flow cytometry? We can't see it by count. You see these 3 are just look different, but not more than that. And so we use the fingerprints. But instead of just looking at it by eye like we did before, we use some computer skills and computer tools to look at it in a more statistical way. Here we see this is outgoing water from the slow sand filter that was old and well working. This is the slow sand filter that had mixed sand, new one. And this is the new one, the red one, that had only new sand. And this is the ingoing water for all of them. So in this plot, we studied the similarities. When 2 dots, which is 2 samples, are close to each other, they are similar to each other. The fingerprint is similar, which means that the natural flora is similar. And when you have 2 dots that are far away from each other, they are not similar. So we see here that the well-established one managed, although we wash it, managed to produce the same natural flora all the time. So the cluster is quite tight. Compared to the one that had mixed one, it's not so spread, meaning that it's not stable, it's changing the natural flora. And this one is even worse, because what you see here, this is the beginning of the washing, and then at the end, it ends here, which means it's actually more close to the ingoing water than the other outgoing water. This is what we can see in flow cytometry. And due to its potential, flow cytometry, many drinking water companies have been really interested into this, and Subatin is really happy to actually have this data over the treatment plants. And I'm really happy to say that we are going to have a workshop and also a training section with with many drinking water companies coming from Stockholm, Linköping, Varberg, to visit our lab. We will open up the lab for them, and we will teach them flow cytometry, because they really want to implement this fast and rapid method into the treatment plant. And that's what we are going to do. And it's due to research and industry. That is due to this collaboration. So I have 1 minute. So I would like to summarize the whole section. We started to discuss about clean water, what is clean water, And then we end, and I gave you some outbreak stories about the football team, for example. And then we started to talk about the natural flora instead of the pathogen and spoilage microbes. And then we discussed a method on how to measure these natural flora. And I show you some methods, and we focus on flow cytometry, and we saw how we use it to go through two different treatment plants, and also how we studied flow cytometry with it. And I would like to end my presentation with a take-home message from this masterclass, and that is that microbes are invisible, yes. They are small, yes, but they play a really important role when it comes to drinking water. Thank you.
You need to stay.
Yes.
Thank you, Sandy, and let me try and understand you better by asking you this simple question. Would you drink it?
Yes, of course.
So there's a whole world in there, but it's a world that we should encourage
Yes.
and embrace. I wanted to also ask you to widen the picture a little bit. We have the 17 new global sustainability goals that the whole world is supposed to reach by 2030.
Yes.
Number 6, and perhaps the most important, and the one that we've been discussing this morning, is to ensure access to safe drinking water for everyone. Are we going to get there?
Yes, we are. We are, yes, believe me. No, I'm a little bit young and naive, yes I am, but I really believe in that by working together, we can reach that goal. We have the skills, we have the knowledge, almost, but we can together do that.
That's such great news, because right now, you know the biggest airplane there is, the A380 that's bigger than the Jumbojet. You imagine filling that plane twice with kids, 2 planes every day, and then you crash them. That's how many people, how many kids die every day from waterborne diseases. So we're a bit off, but we're getting there step by step. And what do you think that we, as a community, as diverse as we are, what do you think that we should do to accelerate this?
We need to collaborate.
And that's what the bridge is about. Thank you again so much.
Thank you.